So the last speaker, oh, there you go. Professor Pratik Shah from MIT, the Media Lab. We put him last because the Media Lab always attracts attention, so please go.
Great, fantastic. I'm the last person between you. There's a panel, and then there's a reception.
No, there is a discussion.
Yeah, I said there's a panel after me, and then there's a reception, so I'll keep it short. I'm going to talk about everything. I'm going to talk about computer science and machine learning, and I know there has been a lot of conversation this morning, why it works, why it doesn't work, where is bias. So I don't have a disclosure slide. I have none. I have a disclaimer slide, which I've put at the end of my presentation about what machine learning is good at and what machine learning is not good at. So I direct a research program called Health 0.0. We're calling it 0.0 because if we called it 1.0, then it worked. But we don't think 1.0 works, so we're just calling it 0.0. So I think I've given several such talks, and I'll let you read this. So I come from the computer science world and have a broad training in biological sciences. So even people who do computer science for a living, there's this massive rat race to do AI or machine learning and succeed to make money or return of investment, help people. But the sad reality is it doesn't work the way it's advertised. Okay, so I'm going to demystify some of it today and then take you on a journey with me on some of my own research that we're doing at MIT. And because I don't have too much time, I always start with the first slide, which is limitations and applications of machine learning. I don't use the word AI too much because we don't think AI exists. We just call it machine learning. So apologies for the AI aficionados in the audience. And there are three basic principles where we use machine learning in the field. And one is automation, which is low-level intelligence. So for example, if you trained a machine learning algorithm with all the images or photographs of all of you in the room, and probably thousands more, and you gave it a new set of images that it hasn't seen before, it will start identifying faces, colors of your shirt, some sort of low-level segmentation. To me, that looks like automation. Okay, it's pretty good at that if you have large data sets, and there is bias, causal inference, inclusion, diversity. I'll buttress my claims as I go along. Okay. The next level up of machine learning is knowledge creation, like how do we use ML to create new knowledge that either humans see, but we don't understand, humans understand, but we don't know how to use it, or humans just don't perceive it. So I'll give you some examples of that. And then the third is actual true intelligence, where an artificially intelligent agent comes up with intelligent cognitive processes that we cannot think of or is as good as us. Okay? Most of the machine learning in the field is automation right now. There are few examples of knowledge creation, and there's very few examples of intelligence. So as humans, we need to be kind to the machine learning algorithms and compare it to where it belongs. That's the takeaway. So I know there's a deep learning revolution going on, but classical machine learning still works. So this is all what we call in the field machine learning. It has been used, you're using it. There is no need to train deep neural networks to do a lot of it. Classification, regression, decision trees, all of those things work very well. Up here somewhere is regularization, rule systems, regression, just standard machine learning. No deep learning, no AI taking over the world. This is what we call machine learning. Okay, so my research at MIT, we look at three separate areas. Some of them are really unorthodox, so I'll introduce them. So the things that we fear from machine learning algorithms, things that algorithms can see but we don't understand, we exploit them for new medical knowledge. We call that machine saliency. The ability of machines to see information in medical images that humans cannot see or perceive or do not use, so we exploit that. We also do unorthodox imaging of biomarkers which only devices or algorithms can perceive. And then we develop novel clinical trial platforms. I'll give some examples in the oncology space for glioblastoma today, to help physicians make informed decisions at the point of care, what to do with the data that they have. And then we collect some real-world data and real-world evidence, which sits outside of the clinical development paradigm with devices and create new digital endpoints for patients. Okay, so as I said, in machine saliency work that we're doing, we we show medical photographs to human doctors, and human doctors say, this is where the disease is. They put a box or a label. Then we run the same medical photograph through machine learning algorithms, and it either segments pixels or information from biomedical imaging devices that humans cannot see or perceive because the photograph needs to be fluorescently imaged or some sort of augmentation from the biomedical device needs to happen. We use those photographs and we combine that knowledge, and we call them neural networks with both human and device cognition. So we take cognition from the human doctors, we take cognition from the medical devices or fluorescent biomarkers that only devices can image. We combine them in machine learning networks, and you can replace the neural network with any classifier. We only use neural networks because they learn fast, we have tons of data, but there is no need to use a machine learning algorithm here. This knowledge can be captured by any learning system. And then we use these combined knowledges to segment out information from medical photographs that gives patients care combinatory, both from humans and devices from simple processes. So we have done some work on that. The second example I want to share with you in the imaging space is how do we exploit machine learning architectures to do unorthodox learning from photographs. So I'll show some examples very quickly. So I think most people in the audience might have heard of staining of pathology slides, prostate cancer, H&E styles. So we got this problem statement from Brigham and Women's Hospital that they had this process where tissue samples, prostate cancer tissue samples, will come to them. They would make slices or take sections, and they would choose 3 or 5 of those slices to stain. And once they are stained, then they would another human would look at it and give medical diagnosis. And this is standard. It seems like you take a tissue, the tissue has to be sliced, etc. So they asked us, is there a way to computationally stain our slides without actually physically staining them, the images? So we've done some recent work on that space. So for that purpose, we use a generative adversarial neural network. What it does, it compares real-world images and synthetic images that we make and starts training a neural network to change the image data in a way that we can synthesize new data. This is not a machine learning conference, but if you want to talk more about our GAN networks, we can talk more about it. And essentially, it starts, there are 2 dueling networks, a generator which generates information, a discriminator at the bottom that fights the generator with a loss function to correct the information, and you get these new representations. So here's an example. So some of the GAN work we have done, which has recently got accepted with Aman Rana and my group, we have taken unstained pathology biopsies, taken slices, taken simple RGB photographs, and then trained these neural networks to computationally stain them. Great. I'll tell you what the value is, right? So that's great. Gee whiz, machine learning research, everybody's happy, but what does this mean? The next problem statement we got from them is that we have already stained many of these pathology slices. They're already stained, but we cannot restain them because they are gone. So we said, okay, so we developed an unstaining network, which takes previously stained photographs, unstains them back to ground truth, like they were never stained before, and then we restain them with the dyes that you want, right? So we have some clinical validation. We are showing this data to humans. We are doing clinical trials. We're going down that road, but I'm just giving you the technology piece right now, right? So we have these neural networks which can stain, unstain, and then aid physicians to use data more wisely, etc. So here are some key examples. So in the machine learning network on the top, you can see the input images, which are unstained. Humans cannot see anything in them, obviously. The target on the second row are actually what humans stain with H&E dye in hospitals. The machine learning algorithm stains them almost identical to what humans stain. And then we have the unstaining example there, where we give it a stained one, it unstains it, and then restains it back, right? So we can reuse data in these loops that way. So then the question is, great. This is great academic research. You have described a way to stain and unstain images. Thank you very much. What does this mean? So right now what we're doing, and we're submitting this to Nature Digital Medicine, we have given these images to human physicians, the only human physicians, I guess, human physicians, and we haven't told them like where these images are coming from. We have given them the images. They think they are H&E images that they see all the time, and they are rating them for tumors, right? So the null hypothesis is they would look at the machine images and toss them out saying, I don't understand what's going on. I cannot find tumors. The non-null hypothesis is that they might find tumors. So keep watching. We'll release that paper soon. We're getting pretty good results with that. Human physicians cannot tell them apart. They're finding tumors in them. So then, great. Now you have an algorithm that stains images. You have a human doctor that has validated it. So how do we deploy this information at the point of care? So as the FDA people spoke this morning, who regulates software? I think Nick, the person before me, spoke software as a medical device. So how do we do clinical trials for these algorithms and integration at the point of care? I want everyone to remember this problem statement was given by a physician to us. We did not go to a hospital with that algorithm saying, give us your data. I just want to point that out. We were happy. The physicians came to us saying that we have this existential problem. Can you make this thing? We gave them the solution, and then the physicians are like, great, now how do we test it? I'm like, I don't know, talk to the FDA. So we are now talking with the FDA about how do we deploy and test these algorithms at the point of care and integrate them only when they are needed and not shove it down people's throats when they are not needed. So that's one piece. And obviously, how do you regulate and communicate this knowledge to a patient or a physician that some diagnosis may be made using machine-generated images? So we're also doing work some NCI and some other hospitals to scale this system, and I'll be happy to talk more about this work. Okay, everyone with me? Okay, great. Okay, changing topics very quickly. So we're also looking at PASH randomized control trials for glioblastomas, prostate, colorectal, and small cell lung cancer. And these are published clinical trial data sets, some of which we have access to. And we're asking this very simple question. Can we redesign these clinical trials right now, looking at historical data? And if we could redesign them with new covariates or machine learning driven knowledge or whatever knowledge, would they look like the trials that were designed by humans before? Okay, very simple stuff. So we're training these general cognitive architectures which learn between linear and nonlinear mapping of data that humans cannot perceive or understand, but algorithms are very good at. Humans are very good at higher level intelligence cognitively, but we are not very good at looking at complex data sets and making linear and nonlinear mappings, and algorithms are pretty good at some of that stuff. So we're exploring that knowledge. So the key example I want to share with you is glioblastomas. I think we are in an oncology forum, so I'll skip the thing. And temolazide and PCV dosing regimens, again, this problem statement was given to us by a hospital. A physician, a clinician walked up to us and said, our patients are experiencing toxicity in temolazide and PCV dosing regimens. Can you redesign these dosing regimens if that's possible? And we said, okay, we'll look at it. So I'm going to skip slides quickly. So we use a reinforcement learning algorithm. This is how reinforcement learning looks in the computer science field. And I think I'll play it. I don't think I have too much time. So it's an automated training agent. It learns, and you give it a reward every time it doesn't drop a ball. Every time it hits the ball and gets a score. Every time it drops a ball, it gets a penalty. Every time it hits the ball, it gets a reward, and it self-learns. Right, it starts learning. Right. And then it figures out after self-learning. So it's reinforcement. It's like penalizing a child. When the child makes a good deed, you give it a reward. When it makes a mistake, you penalize it. This machine learning does not work in medicine. Okay, this is not the machine learning we should be doing in clinical work. Okay, this is the reason why computer science probably has a bad reputation in medicine. The problem is, as Giovanni pointed out, these require tons and tons of data, the simulators. It's a deep neural network that Google had access to, and we trained it. We don't have access to the data. So we had to do unorthodox machine learning to solve this problem. So we designed an environment where an artificially intelligent agent, this is borderline AI, not ML, artificially intelligent agent, every time it dosed or gave a dose was penalized. And every time it achieved a drop in the size of the tumor, it got a reward. So we made it compete in an environment to maximize decreasing the size of the tumor while still giving least amount of dosing. Okay, I don't have too much time, only 5 minutes left, but I'll talk more about it. And the way we forced it, since the Board of Mathematical Sciences is sponsoring this, I had a mathematical slide. The way we gave it the reward was we told the reinforcement learning agent, every time you reduce a tumor, we will give you a small reward. So RMTDT minus RMTDT plus is the time step where it reduces the tumor. But we'll subtract the penalty, which is the amount, multiplied by the concentration of the drug you gave the patient, right? And eventually we give it a final reward saying that we don't care what you do, but at the end of your training, if you don't reduce the size of the tumor, we don't care about you, okay? So broadly. And we gave these hyperparameter searches, etc. So again, this is how humans, this is from the data that we got from the hospital, this is how humans administer temolazide to patients. The two blue lines are the start and the end of the trial. The size of the tumor keeps going down, and the black horizontal lines are the dosing. So the null hypothesis was that the machine learning agent would say, this works. So the non-null hypothesis is what the agent came back and said, that for the same cohort of patients, if it had to make one policy, a single policy, to treat all of them, it would not use the same number of doses that human experts would use. It would stagger the doses and give them at different intervals and skip doses. Okay, this is simulatory data. Okay, so this is the first policy. Because this is reinforcement learning, we have policies. We don't have AUCs and ROC curves. These are actual policies that the machine learning agent makes. Right, so if there's a patient enrolled in this clinical trial, humans would have given that patient a dose by PK/PD calculations, which is great, every month because it hasn't been de-risked. Some of the algorithms we are showing that for those same cohorts of patients, many doses could have been skipped. Okay, so we'll again get to deployment, clinical trials, FDA regulation for this work. The next question we asked this machine learning agent is that if it's a cohort-based trial and if all of us are in the trial, it would, humans would give the same dose which is optimized by early preclinical work. So we gave the machine learning agent the flexibility to say, do you have to treat all these patients with the same dose? It said no. It decided to give individual patients different doses. And those doses were designed by the covariates of the biomarkers on which the model was trained on, right? So there's some early examples of personalized or deep personalized machine learning for clinical trial work that we're doing. And it again got reduction in tumor sizes. Okay, so I have 3 minutes. Then we gave the AI agent another flexibility. When you see a patient, do you have to give the full dose, half dose, quarter of the dose, or no dose? And at every time, it came back with a new dosing regimen, which used full doses in some patients, quarter doses, half doses. So this is standard risk-reward machine learning work that we are doing. The reason it cannot be unsupervised is because it needs some guidance from humans where it should not just optimize rewards and just keep dosing patients. Okay, so I have 2 minutes, 33 seconds. So I'll wrap it up. The third area that we are looking at is real-world data and real-world evidence. And I think there's been a lot of conversation. I've been reading a lot of reports, and I'm thoroughly confused about how real-world data, real-world evidence, EMR, EHR, EPIC, and everything that people are talking, how do we integrate that to benefit patients? So we take a very simplistic approach in this field. We are just trying to look with imaging, paper diagnostics, all sorts of wearable devices, novel endpoints outside of clinical trials that could allow patients to be seen more quickly and robustly in the real world, right? And we're also doing some work in EMR and EHR records with machine learning algorithms, and I'll talk about it later in the reception if people want to talk to me about. But I think the real problem with real-world data and evidence is that we are stuck with the evidence that has been collected. We are trying to create new endpoints and collect new evidence. So classic examples are biomarkers, which can be imaged on mobile phones, paper diagnostic strips, which can be deployed at home and change colors, and stuff like that. Okay, and we published this paper in the BMJ where we showed that the value of these things compared to primary care is a better stratification of patients, getting them care quicker and faster, and maybe informing clinical trial processes to conduct these real-world evidence generation studies at the point of care to supplement trial results. Okay, this is some of my publications. Since I'm an academic, those are my disclaimers. There you go. Okay, I have 57 seconds. Okay, so what does this mean to you in the audience? If you're an oncology specialist, if you're a foundation, if you're a patient, and you hear about machine learning, people are criticizing it. I'm talking about what you do. So we have created this new data and research sandbox at the MIT Media Lab, and I think there's some familiar faces here, where we are aggregating this knowledge from our experiences in the field and trying to create collaborations between academia, government, life sciences, and everyone to look at machine learning for what it's good at and what it's not good at, make sure that the data is not biased, and hopefully that will lead to new models and technologies. For those of you who want tools, we do a variety of approaches with training with less data, training with labels, learning to learn. How do you know what to learn? So meta learning and stuff like that. And so we are doing this research program called Health 0.0, which I'm leading. And final thing, in 1 second, if I get permission, I want to end this with language and literacy for machine learning research. So inclusion and diversity in data sets are key. If you have a biased data set, and if you're a machine learning person or oncology person, you have a biased clinical development process, which is going to come along again. So we have the opportunity to develop a new clinical development paradigm with more inclusive and diverse data sets. We look at causal inferences and not just correlations when we train algorithms to make sure that what we are seeing is truly a cause and not a correlation that was discarded as part of the data. We don't think in computer science and in my group that there is a competition between humans and algorithms. We think they should work in synergy in the next 5 or 10 years. I don't think machine learning is there yet to take any jobs. We do think that black box phenomenon of machine learning, explaining the algorithm is important, but it should not be rate limiting to create new knowledge to help patients. We should not be using that as an excuse to give good technology out of the hands of patients. New clinical trials for algorithms need to be piloted because we don't know how to regulate some of this stuff. And more importantly, regulation and communication with patients and physicians is important. It's a nascent field. Rather than invention, we need to de-risk it and explain it. And understanding the difference between automation, knowledge, and intelligence. And as I said, it's a new field. We can do it right together versus operating in silos. So these are the people who make it possible, my students and my staff, collaborators, institutions, and thankful to Joey Ito, the director of the Media Lab, who supports and mentors me for some of this work. Thank you.